Good day to our instructor, Dr. Janelle Mark D. Sarno. In this video presentation, we're going to discuss on how can DNA profiling be of help to criminal investigation. So first, what is DNA? DNA is also known as deoxyribonucleic acid is a chemical substance found in all cells of living organisms whose composing have been passed on forms of parents to offspring. It is called as the genetic or hereditary material. A person's DNA is the same in every cell. Chemically, it is an acid and it is composed of three subunits, namely, the first is the phosphate group, second is a deoxyribose sugar, and last one is one of the four bases. Also, it is composed of adenine, the A, thymine, the T, cytosine, the C, and last one is the guanine, is G. The human body contains 100 trillion cells. There is a nucleus inside each human cells. Each nucleus contains 46 chromosomes arranged in 23 pairs. One chromosome of every pair is from each parent, so 23 for mother and 23 for the father. The chromosomes are filled with tightly coiled strands of DNA. Genes are segments of DNA that contains instruction to make proteins and the building blocks of life. Now let's proceed to the discovery of the DNA. In 1953, DNA, the so-called building block or the genetic blueprint of life, was characterized by the scientists Sir Francis H. Crick and Sir James Watson as the material that make up the genetic code of all organisms. Crick and Watson were completely unaware of the potential impact of their discovery could have on forensic science in particular and the criminal justice system as a whole, resting on the established natural truth that no two individuals has the same DNA blueprints, with the exemption of identical twins. DNA typing or profiling, as it is now called, was conceived. In 1984, DNA analysis was first recognized as having application to forensic science by the British molecular biologist Sir Alec Jeffreys. The thereafter directly used in a rape and murder cases to exonerate an innocent suspect and to convict the guilty accused. Sources of biological evidence are blood, semen, saliva, urine, hair, teeth, bone, and tissue. General characteristic of genomic DNA. Each person has a unique DNA profile except on identical twins. Each person DNA is the same in every cell. DNA from skin cells will match from blood cells. An individual's DNA profile remains the same throughout life. Half of your DNA comes from your mother and the half from your father. DNA analysis, a new technique. Why we use it? DNA contains detectable patterns unique to each individual. DNA is a robust molecule and is stable. DNA can be isolated from a wide range of biological samples. Source of it's the same in all sources like blood, semen, sputum, skin, and etc. Precise pattern can now be detected even with very small samples like blood spot, single hair, follicle, lip prints on a glass, physical fingerprints, saliva, skin on a cigarette butt, and etc. Line up of cases where DNA analysis can be of help. First is sexual assault cases like rape, murder, homicide, robbery, hit and run, extortion, paternity cases, identification of remains in mass disaster, fight against human trafficking, especially for kids, prevention of illegal adoption of kidnapping, stolen or trafficking children.
And now, let's proceed to the collecting of DNA evidence. So, in collecting DNA evidence requires strict adherence to protocols to ensure integrity, prevent contamination, and preserve the chain of custody. So, here are the standard guidelines for collecting DNA evidence. The first one is the preparation. So, you must wear protective gear such as gloves, mask, gown, and hair cover to prevent contamination. Use sterile equipment, single-use or sterilized tools such as swabs, tweezers, scissors, and label materials clearly before collection. So the second one is identifying DNA sources. The common source include blood, saliva, semen, vaginal fluid, skin cells, for example, clothing under fingernails, and hair with follicle, touch DNA, for example, from weapons or door handles. So the third one is the collection techniques. So in swabbing, moisten sterile swab with distilled water if dry stain. Then col- collect samples in a circular motion. Cutting. So cut out stained areas of fabric if swabbing isn't ideal. So lifting. Use tape for dry skin cells or trace evidence. And the last one is control samples. Collect a substance control or unstained area nearby to compare possible contamination. So the fourth one is preservation and packaging. So air dry all wet samples before packaging and use paper containers, not plastic, to avoid mold growth. And seal and label each item with case number, collector name, date and time. And avoid cross contamination. Use fresh gloves and tools for each sample. So the fifth one is documentation. Record location, type of samples, method of collection, and conditions. Maintain detailed chain of custody documentation. So the last one is transport and storage. Store in a cool, dry environment. Transport to the lab as soon as possible. And long-term storage, frozen or in low humidity environments depending on sample type. And now, let's proceed to packaging and transporting DNA evidence. So, in packaging and transporting DNA evidence must be done with great care to preserve its integrity, prevent contamination, and maintain the chain of custody. So, here are the detailed guidelines. So, the first one is the general packaging guidelines. So, it's use appropriate containers such as paper bags or envelopes for biological evidences, just like blood, saliva, and etc. For cardboard boxes is for larger items like clothing, weapons, and etc. And plastic containers only for dry and non-biological items used with caution can retain moisture. And the second one is to avoid plastic for wet or biological samples such it can trap moisture and promote bacterial growth, degrading the DNA. So, air dry. Air dry wet items completely before packaging. Separate items individually to prevent cross-contamination. Use separate containers and label each item clearly. Seal all containers with tamper-evident tape and label with case number, item number, description, date and time, collector's name or initials. The second one is chain of custody documentation. So, log every transfer of the evidence, just such as date and time of collection, name of person collecting, location and description of the item. Every individual who handled the evidence afterward use official evidence forms or logs to track this info. So the third one is transporting evidence. So use clean, secure transport containers for lockable evidence boxes or coolers. So avoid extreme temperatures. Transport in climate-controlled conditions if possible. Do not leave evidence unattended in vehicles or unsecured locations. Deliver directly to the forensic lab as soon as possible. The fourth one is special considerations. For liquid biological samples, store in sterile containers, examples test tubes with secure caps, and keep refrigerated at 4 degrees Celsius or frozen if delayed expected. For DNA swabs, place in sterile envelopes or swab boxes, allowed to dry completely before sealing. So that's for the packaging and transporting DNA evidence.
Now, in this video presentation, our instructor instructed us to find a criminal case that has been solved through DNA. So we found out the first criminal case in the Philippines that has been solved using a DNA testing. This was the case of People versus Vallejo y San Martino, or case GR number 144656. The incident and crime. On July 10, 1999, in Barangay Ligtong 1st, Rosario, Cavite, the accused Jericho Vallejo y San Martino was charged with the rape with homicide of Daisy Jolola, a 9-year-old child. It is alleged that while with the victim, the accused with law design and by means of force and intimidation engaged in sexual intercourse with her, and during the act strangled her to death. The crime occurred in a context where the victim was at her neighbor house for tutoring and the accused residing nearby at Miss Vallejo's house. Later accompanied Daisy under the pretext of retrieving a book. Chain of event and initial discovery. The victim, Daisy Jolola, was sent by her mother, Nida Jolola, to the house of their neighbor for tutoring by Aimee Vallejo, the sister of the accused. Daisy returned later with the accused, and the subsequent event led to her disappearance. Nida spent the rest of the day searching for her around 10 a.m. in the morning. The following day, Daisy's body was discovered tied to the root of an aroma tree by river at the barangay hall. Witness testimonies and circumstantial evidence. Multiple witnesses testified regarding the victim's last known whereabouts. Nida Jolola testified that Daisy left with the accused after visiting Aimee Vallejo's house. Jessamine Mataverde and Charito Paras Yepes observed the accused in the vicinity. Jessamine noted that Daisy was seen playing outside and then later accompanied by the accused, while Charito testified about the accused's unusual behavior near the seashore, including his wet clothes, which contrasted with his face and hair. Barangay official and police details. Upon receiving report about the missing child, barangay officer and police promptly taken the accused into custody. The accused clothing, a white basketball shirt with printed San Martino, and violet basketball short marked with number 9 were recovered. These garments were blood-stained and later submitted to forensic examination. Forensic and medical evidence. Forensic findings by Dr. Antonio S. Vertido during the physical examination of the accused revealed several abrasions, contusions, and hematoma. and a laceration on his left ring finger. The autopsy of Daisy Jolola showed sign of post-mortem decomposition, multiple contusions, abrasions, lacerations, and nail mark on various parts of the victim's body. Specific finding consistent with manual strangulation and sign of sexual assault, including injuries to the genitalia. Bloodstain analysis, forensic biologist Pet Byron Buwan's test indicate that the bloodstain on the recovered clothing from the accused and on the victim's clothing were positive from the human blood. Both testing with group A characteristics, DNA test on a vaginal swab of the victim confirmed the presence of the accused DNA profile. Confession and extrajudicial statement. The accused during various stages of investigation made several admissions. Initially, he admitted to killing the victim by strangulation when interviewed by Mayor Renato Abutan. Corroborated by the latter testimony in the subsequent encounter with NBI forensic biologist Pet Byron Buwan. The accused confessed in the presence of legal counsel, Attorney Lopo Leiva, and police that he raped and killed the victim. A handwritten extrajudicial confession was also produced. Executed by the accused in his cell, the confession reiterated his admission of rape and homicide. The accused claimed to have executed and confession after being allegedly maltreated by police. Through his confession was given in the presence of counsel and later corroborated by prior statement made to other officials. Defense presentation and additional development. The accused and his sister Amy Vallejo testified in defense. They provide an alibi stating that on the day of the incident, the accused has been at their residence and was engaged in drawing a project for a school assignment. The defense contended that the accused statement to the police were the result of coercion and maltreatment. The defense further challenged the handling of evidentiary matters issued were raised regarding the integrity and probative value of bloodstained garments and the DNA evidence. The defense also questioned the admissibility of both the oral and written confession, arguing that they were obtained under duress. Sentence rendered on the accused. The accused appellant is Jericho Vallejo Isa Martino. He was charged with rape with homicide and sentenced to death by the Regional Trial Court. On appeal, the Supreme Court affirmed the conviction and death sentence, finding that the circumstantial evidence and DNA evidence were sufficient to establish Vallejo's guilt beyond reasonable doubt, even without considering his extrajudicial confession, which he claimed were coercive.
How does DNA profiling can be help to criminal investigation? So, DNA profiling can help criminal investigations in several important ways. The first one is identifying the criminal. DNA left at the crime scene, such as from blood, hair, skin, etc., can be matched to a suspect, helping to confirm their involvement. The second one is clearing the innocent or elimination. If a suspect's DNA does not match the evidence, they can be ruled out, which prevents wrongful convictions. The third one is linking multiple crimes. If the same DNA appears at different crime scenes, investigators can link the crimes to a single perpetrator. The fourth one is identifying the victims. In cases where bodies are unrecognizable, DNA can help confirm the identity by comparing it to relatives. And the fifth one is cold case resolution. Old cases can be revisited with modern DNA technology to find new leads or solve long-standing crimes. The last one is providing court evidence. DNA is a strong form of forensic evidence and can be used in court to support prosecution or defense. That's all. Thank you.